Electrophoresis is a laboratory technique used to separate biological molecules based on their size and electrical charge. An electric current is used to move molecules to be separated through a gel. Hi, I am Manang Biday, and in this video, let's take a step-by-step on how to construct an electrophoretic setup using simple materials, and you shall be able to see how it separates samples, in this case, food coloring. First thing that we need to do is to assemble the materials needed for this experiment. First, we assemble a gel comb similar to this figure. The comb is made out of 5 coffee stirrers evenly spaced from each other, having the same height from the bottom of the box. They are then sandwiched in between 2 popsicle sticks. We tape them all together and the gel comb is formed. Next, we prepare the gel. 50 ml of 1% weight per volume colorless gelatin is prepared in a buffer solution made of 250 ml 0.2% weight per volume of sodium bicarbonate in distilled water. The resulting solution shall be in gel solution. It is heated and stirred several times until the gelatin powder is completely dissolved. The final solution is translucent in color. The comb is now positioned 3 cm away from the wall of the plastic box. The gel solution is poured into the box until 0.5 cm of the teeth are submerged in it. The solution is then left to cool and solidify. After the gel solidifies, the comb is carefully removed, and the gel is cut 2.5 cm from top and bottom. The extra gel is discarded. Installing the electrodes is the tricky part. The wires are bent such that both fit inside the box and can be hooked at the sides. After this, the buffer is added until the entire gel and its wells are submerged about 3 to 5 mm on top. The samples are then prepared by mixing 1 ml of glycerol with 2 drops of food coloring sample. Using the droppers, the samples are loaded into the wells. Five 9-volt batteries are clipped together. The positive terminal was connected to the electrode at the bottom of the gel, while the negative at the top. A closed circuit is now formed, thus the setup is now complete. Yay! Now, let's observe what happens. Our setup took a little longer than expected, but it took us 2 to 3 hours to see visible results. As we can see, none of the samples moved towards the negatively charged electrode, which means that the dye molecules which were loaded into the gel were negatively charged at neutral pH. The yellow, red, and blue dyes migrated to the positive charge, with the yellow dye traveling the farthest. Moreover, the blue and yellow components from green were separated, as well as the blue and red from violet. By conducting an electric current through an electrolyte buffer, like the sodium bicarbonate buffer that we used, charged molecules will migrate towards a terminal with the opposite charge. Once suspended within the polymer matrix, such as the gel, the molecules will move at different rates based on their size. The smaller molecules move faster than the larger ones since they are able to pass through the pores of the gel easier than the larger molecules. In this case, we added glucose to all samples so that their relative sizes and masses were equal. The rate of migration would now depend on the charges of dyes. As mentioned earlier, the yellow dye traveled farthest towards a negative electrode, so it has the most positive charge among the dyes, followed by red and blue. Separation of colors was also observed, as shown in the wells of green and violet. The colors then traveled according to their charges. Gel electrophoresis is a very essential tool used in molecular biology and genetic engineering. This experiment demonstrated the ability of gel electrophoresis to separate the mixture of dyes into their individual components by the application of a combination of dyes to the same sample well.